Well, welcome everybody. I'm here to talk to you about the launch of the Biometrix Technology Suite. This is specifically focused on the Neuro MRI, and I'm really excited about finally being able to get this out the door to everybody. I've been working on this for over 3 years now, so 3 years of work is coming to fruition into what I think is the most comprehensive and most usable MRI technology currently available today anywhere on the planet. So the Biometrix Technology Suite is really designed to work integrated with each other. So the Neuro Restore, which is a comprehensive nutrition package that is specifically designed to restore the physiology and structure and function of the human brain, as well as your heart and other parts of your physiological system. And it's really designed, and you're going to see in this presentation today, how it actually works to restore brain structure and function. The Biometrics Bioscan, which many of you are familiar with the proton scan, this is an expansion of the proton scan technology that now uses the full power of the ion cyclotron non-targeted platform. It actually measures over 11,000 biomarkers on the mass spectrometry platform in combination with a bunch of other clinical chemistry parameters. And what we're going to talk about today really is the Neuro MRI, which has been over 3 years in the working, understanding the currently available technology of MRI and what is really available for advanced doctors like yourselves is very limited. Most people can only get volumetric analysis, which can be quite misleading and quite frankly, useless in many applications. And so understanding the structure, the function of the human brain using advanced MRI is critical in the work that we're doing for reversing aging and restoring brain health. So the Bioscan key highlights, we've really expanded the ethanolamine platform. Many of you, wear the plasmalogens, but we've added the omega-6s, omega-9s into the plasmalogen pathway, the ethanolamines, the full phospholipid indices. These are the indices that are published in the paper that we did several years ago with Dr. Jordan in Santa Monica. And then also added the lysoplasmalogens, which are part of the phospholipase A2 system. Choline plasmalogens have been expanded as well. Phosphocholine system added the complete phospholipid indices as well as lysophospholicholines. There'll be a whole other presentation on the utility of all these markers. But elongase, gastrointestinal tract acids, related to cancer, colon cancer, pancreatic cancer, mitochondrial function testing, both in the ethanolamine pool and the choline pool. Quick measurement of iron. But the methyltransferase and choline system are the same, sphingomyelins and ceramides, but we've really dramatically expanded the sphingomyelin ceramide area so we can actually look at the individual species, because some of these species have opposite risk factors, especially when it comes to cardiovascular disease. Inflammation and oxidative stress panel has been dramatically expanded to, mostly the GTAs, the C-reactive protein, but we've added malondialdehyde, catalase, superoxide dismutase, galectin-3, and cortisol measurements into the Bioscan measurement panel. And then peroxisomal function, looking at, fasting triglycerides, of course, glucose regulation, cholesterol transport, kidney function, creatinine, creatine kinase, blood urea nitrogen, uric acid. So much more comprehensive, added a few critical components to it, but really, if you want to understand human biochemical health, this is where you want to start. So onward and upward to the Neuro MRI. So what is this all about? It's the world's first and only commercially available complete digitization of the human brain. And this is a quantitative, objective analysis of human brain structure and function. So what is it? It's complete. Neuro MRI generates a complete biochemical, structural, and functional assessment of the human brain. Most importantly, it's quantitative. Neuro MRI is quantitative, which enables you to objectively track and evaluate and optimize evidence-based interventions designed to restore and maintain optimal brain structure and function. It is life-defining. The beginning of your perpetual health journey starts here. There can be no healthy longevity without brain health. And you're going to see data about that in this presentation. Why Neuro MRI is your most valuable health asset is that the structure and function of the human brain is irreplaceable. Okay, I can fix your arms, I can fix even your heart, okay, but I can't fix your brain. Learn it, protect it. If you do not track the structure and function of your brain, you are aging in ignorance, which none of us want to be. Important thing is that there's validated protocols that objectively improve brain health now. Okay, these now exist. Every person, regardless of age, needs a baseline neuro MRI. There is no other way of knowing the true status of your brain. Accidents can and do happen. Know what your brain looks like before you have an accident and financially protect yourself. I can't tell you how many times people come in after an accident saying, show me what happened. Show me what happened in my brain because I've got a concussion from a car accident. Well, it's very difficult if I don't know what your brain looks like before the car accident. So get your baseline MRI done, store it for future protection. NeuroMRI is focused on these core components of the human brain, and we measure this using multiple different sequences in the MRI. We look at basic volumetrics, which most people are familiar with, cortical thicknesses, which is actually more important than the volumetrics. We look at axon and dendrite density, inflammation of the brain, the structural connectivity, the functional connectivity, the blood flow, and the biochemical composition. And all this information is correlated and combined to give you a complete picture of the individual structures of the brain, how they're physically composed, and how they're actually interacting with other parts of your brain. So let's start. Why is it so important to do this? What is, why are we focusing on the brain versus the heart and the lungs or your kidneys? And the point is, part one is talking about the shrinking brain of ours. And the brain shrinks. Okay, this is the brain volume with age. And this has been studied over and over and over again. It's a highly reproducible observation that the total volume of the brain shrinks. Obviously, your skull doesn't shrink. Your head looks the same when you're 80 as it did when you're 40, but what's inside your skull changes. And what it does is it shrinks, okay? And as it shrinks, your total gray and white matter decrease, and the cerebral spinal fluid, which is basically the water in your brain, increases. And why that's important is that the shrinkage of your brain determines how long you will live. So this has been studied over and over again. So this is looking at brain volumes at age 70 and all-cause mortality going forward. So depending upon the size of the brain at age 70, which basically means how much has your brain shrunk from age 40 to age 70, right? Assuming most people at age 40 all have healthy brains. So some people have aged kind of age 70 to have relatively large brains, remain brain volumes, and some have shrunken brains. Those who have shrunken brains by age 70 die much quicker than those who do not. Now, we've studied plasmalogens as a critical component. Many of you will already be aware that 10, 20, 30, even more percentages of the brain volume is actually these critical lipids called plasmalogens that your body must make. There's small amounts of it in human breast milk. And we have now advanced nutrition that can restore plasmalogen levels across the entire human body. But what we do notice is that in the general population, plasmalogens typically peak in our late 40s, early 50s, and decline quite precipitously after that. This is in humans. And if we take a look at animal models of animals who have a plasmalogen deficiency, these animals have reduced white matter and they have shrunken brains. So we know the level of plasmalogens in the human body is highly related to brain volume and brain quality. What we didn't know until a few years ago was how important your blood plasmalogen levels are to your overall lifespan. And so I've talked about this pretty extensively. We're talking a 30-year differential in 5-year mortality based upon the levels of plasmalogens in your blood. So brain volume and plasmalogen levels are a critical component to understand and to get right if you want to live a long and healthy life. So how do we measure brain volume? So we're looking at the brain itself, the core structures of the brain. People understand there's gray matter and there's white matter and there's cerebral spinal fluid. Okay, the gray matter is this outer band of cells in your brain called the cortex, if you will. And then the white matter falls in between them, like the inner white matter, the corpus callosum, which is basically a bridge between your two hemispheres. And the ventricles is where you have cerebral spinal fluid. And we look at it on an MRI, how an MRI shows this information is that this little band of cells on the outer surface of the brain, which is your cortical surfaces, is called gray matter. White matter is on the inside. Corpus callosum, which is a white matter bridge between the hemispheres. Cerebral spinal fluid shows up as black here, and the ventricles are areas inside your brain that contain cerebral spinal fluid. And so when we look at these core areas of the brain, what typically happens is using high resolution volumetrics, we notice that the gray matter will decrease with age. This white matter also decreases with age. So this is general brain shrinkage. And as the gray and white matter shrink, the cerebral spinal fluid increases to fill up the empty space. So this increases with age. And then using volumetrics, we can identify regions of interest. We can identify the hippocampus, the amygdala, frontal cortex, so on and so forth. So brain volume changes with age. What's really going on here? And this has been studied extensively, as you can imagine. So this is looking at the different lobes of the cortex, and this is the average rate of shrinkage per year. And so what you're dealing with here is that on average between 0.3 and 0.5% of these areas decrease every single year of your life starting at age 40. And this is a general picture of what a brain looks like with your frontal lobe, occipital lobe is back of your head, temporal lobe is by your ears, parietal, and so on. And so when they look at this and they want to quantify exactly how much does the brain shrink on average as we get older. And from age 40 to 60, we're dealing with roughly a 14% loss of gray matter, 11% loss of white matter, corresponding to about a 36% increase in cerebral spinal fluid. And your ventricle space almost doubles in size, okay? 87% increase just from 40 to 60. And it gets worse from there on in. And so what we measure with the Neuro MRI using high resolution volumetrics, we measure 98 specific cortical gray matter volumes, 19 subcortical gray matter volumes, 34 cerebellum volumes, 53 cortical white matter volumes, 7 cerebral spinal fluid ventricular volumes, and then there's certain combinations of volumes that are added together. In total, there's 253 total volumetric regions of interest measured in the Neuro MRI. So part 2 is the thinning brain. So you want to measure your volumes because those are gross, crude measurements of brain aging. The thinning brain is the other part that many of you may be familiar with. And the brain has this band of cells on the outer surface of the cortex, and that's where most of your gray matter is. And so there's outer surface and then the inner surface. So cortical thickness measures the thickness of that band. And this outer band is not just one cell layer. There's actually 6 cell layers. And it's fairly complex because there's communication happening in between these cell layers. So for example, when you hear something or you have touched something, you get an efferent or an afferent fiber will come into the brain and it'll connect in one of these regions. And then this afferent input will then transfer its information to another layer of this band, and that'll give you your efferent level. So if I want to feel something hot, then move my hand. Okay, there's a spinal cord reflex, but a cortical reflex would be the heat would come in at one layer, and the movement of my hand would be moved from another layer. So this cortical thickness is very, very important. It's not just some band of cells. It's 6 layers interacting with each other. And so when this cortical thickness shrinks, the actual functionality of the human brain is dramatically affected by the thickness of these cortical regions. This cortical thickness reproducibly decreases with age. As we get older, the thickness of that gray matter band gets smaller and smaller. And if we look at the importance of this in human function, so one of the core functions of the human brain is cognition. And so Alzheimer's disease or loss of cognition is a critical variable in understanding how is that brain functioning. And so we look at young onset Alzheimer's disease. These are individuals at age 60 looking at brain thickness as well as some advanced technology called neurite density. And what you find is that at the same age, so these are age-matched individuals, healthy controls versus young onset Alzheimer's disease. Those with Alzheimer's disease in their 60s have reduced cortical thickness, significant thinning of the cortical bands. Neurite density, which is called NDI, is also dramatically decreased, and orientation dispersion index, which is another measure I'll get into later, also decreases slightly in aging. So persons with dementia have reduced cortical thickness. I'll talk about this NODDI values in more detail a little bit later on. But if we correlate cortical thickness specifically with mental test scores, so MMSEs, for example, regardless of whether you have dementia or not, cortical thickness correlates with your cognitive function. So a positive variable of 0.47 means the thicker your cortex is, the better you perform on the mini mental state exam. The greater the density, neurite density, the more dense that cortical band is, also is associated with increased cognitive functioning. Now, we published this at the Alzheimer's Association several years ago, understanding what is the relationship between the plasmalogens in your blood and the thickness of your cortex. And this study showed very clearly that those individuals with decreased blood plasmalogens were associated with thinning of the cortex, and especially in here, this is your temporal cortex, and this area here is in your hippocampal regions. Very specifically, the relationship between blood plasmalogens and cortical thickness is directly related to those areas most involved in cognitive functioning of humans. So cortical thickness is a big deal. We want to know what's going on. You want to know where your cortical thickness is and where it's going. So Neuro MRI measures 600 cortical surface regions, 300 per hemisphere. So a very comprehensive surface map of the human brain, measuring the thickness in all of those regions. And it's organized into 17 networks. So you can look at different areas of the brain that are thick and thin. And now the brain will have a natural distribution. So not all areas have the same thickness. So some areas will have a naturally thinner area, and some areas will have a naturally thicker area. And that's why comparing to a population is important. And you'll get a histogram analysis of all 600 regions and how many are in what bin of thickness. So that's why we want to measure cortical thickness. Is a very important thing to measure. Now we get into the real details of how the human brain really looks like and how it really functions. And this is the denervating and inflamed brain. Many of you will be familiar with inflammation of the brain and understand, obviously, an inflammation of the brain causes reduced function, brain fog, all these issues. So if you're seeing a symptomatic effect of brain inflammation, that inflammation is actually being translated into some level of denervation. So inflammation and denervation go hand in hand. Multiple sclerosis is a very good example of that. This is where more advanced technology comes in. This neurite orientation, dispersion, and density imaging. It measures brain structure, not volume. And what it does is it takes every three-dimensional voxel, so of the brain, the MRI machine measures things in a spherical, in a cubical fashion. And so it takes every cube of the brain, it divides that cube into three types of water. It can measure the intraneurite water, so the cellular water. It measures the water that is actually in fibers. And the fibers of the brain are defined by your axons and your dendrites. So axons are what send inputs from a neuron, and dendrites are what sends inputs into a neuron. And then the second cellular type of water, it measures water that's inside a cell body. This can be a microglial cell or a neuron cell or an oligodendrocyte cell. So it's still a type of water, but it's in a cell body, not in an axon or dendrite. And the third part of the water it measures is what's called free water, water that isn't constrained by any cellular barrier at all. So by looking at these three things, you can get a very, very powerful understanding of what every single voxel of the brain is comprised of. You're getting a detailed structural analysis of the brain. So NODDI measures three independent water components of a 3D cube: intra, extra, and free water, which always add up to 1 because you have a cube. Something's got to be there. There's no air in your head, right? It's either cerebrospinal fluid or it's tissue. So it always has to add up to 1, which means changes in one component is always reflected in changes in the other two. And by measuring these three components, you get a very clear understanding of how each structure of the brain is performing with time. So as you can imagine, white matter and gray matter will look different. Okay, so this is a measurement of the corpus callosum, for example, and the corpus callosum is this major fiber tract that connects the two hemispheres. It's a big bridge between the two hemispheres. And it looks like this. It's a bunch of fibers. Those are axons, and then the myelin sheath is contained around that axon. And so this neurite density, this intrawater, is really a measure of myelination. Okay, so cells that are not myelinated don't show up as intraneurite water, they show up as extraneurite water. So really, the intraneurite water is really myelination measurement. And so lots of myelinated fibers in this white matter tract, and there's a small number of cell bodies, okay, typically like your oligodendrocytes. So in these very dense white matter areas, you're going to see high levels of intrawater and relatively low levels of extraneurite water and relatively low levels of free water. That's what a good white matter looks like. Now your gray matter, which is this band that we talked about for cortical thickness, for example, that's a whole different story. It's a very complex situation. We're dealing with 6 layers of neurons interconnecting with each other. So now you're going to have both axons, okay, and dendrites, and these cell bodies. So you're going to have a much more complex picture. Okay, so the neurite density is going to be much lower because there's a lot more cell bodies. So a healthy gray matter structure looks something like this. There's a mixture of neurons, cell bodies, and a mixture of axons and dendrites. And then they should be in a relatively proper proportion. What happens when the brain gets inflamed, though, this is where the NODDI values really help us a lot, that you can't really get by any other measurement in the human brain. This is the only way we can actually do this. So if you take a look at this, is an example of an animal study looking at stroke. So it's an occlusion. They block the blood flow to the brain of an animal and wait for a stroke to happen. And then they wait, and the brain's response to a stroke is inflammation. And inflammation in the brain is called microglial cells. And so normal hippocampal system looks like axons and a small number of cells around. But as the brain gets inflamed, the number of these microglial cells dramatically increase. And that's going to show up as a change in the NODDI variables. So increased cellular extraneurite water. Inflammation also causes demyelination and decreased cellular intraneurite water. So if we look at this, as a variable, so if microglial count, so there is studies been showing that as microglial levels go up, your microglial density is positively correlated with this extraneurite space, which is the second variable in the NODDI analysis. And if you look at a classic disease of inflammation like multiple sclerosis, and you look at this V, so this V in means the volume of intraneurite water, okay, just a short form. And you can see very clearly here that in people with multiple sclerosis, there is widespread decreased levels of intraneurite water. And this is caused by the inflammation of the brain. So the inflamed multiple sclerosis brain is reducing the myelination, which is resulting in reduced neurite density. And you'll notice that it's not just in the lesions, it's across the entire brain. No multiple sclerosis person has a brain that looks like that on T2 scan. Okay, that's not going to happen. Okay, you're going to have spots in different spots, but what you're seeing with neurite density imaging is that normal appearing white matter has reduced density as well. And so NODDI is a much more powerful, much more sensitive measurement of the effect of inflammation on the entire human brain. So reveals widespread axonal denervation in multiple sclerosis, normal appearing white matter, and in the MS lesions. So that's an inflammatory disease process. But what about just general aging for people without multiple sclerosis? Well, this has also been studied very extensively. So here's a paper that looked at a huge number of variables that are associated with the changing of the brain structure as we get older. And what they found was, of all the variables, first of all, you'll take a look at this age plot here. You'll see how the brain sort of matures in the age 40 to 50 range. It seems to reach its kind of maximum functionality. And then the wheels come off the bus, okay? And so all of a sudden, the brain starts changing quite dramatically after age 50. You can see just on this plot. But the most sensitive change with age is this intraneurite water volume fraction. So this NODDI intraneurite. And this, but you'll notice that this increase in free water doesn't exactly match. If the free water increase was related exactly to this fiber density, then you'd expect this free water to be way up here as well. So it should be mirror images of each other, but they're not. And that's an important distinction to understand. What's so powerful about this intraneurite water from NODDI is that it's impossible to misinterpret this variable. Okay, it is purely measuring the axon and dendritic density of the three-dimensional space. And what you can see here over the course of age, from age 20 to over 80, again, you see this peak neurite density happening in our 40s and 50s, and then this precipitous decrease in fiber density as we get after age 50. Axonal and dendritic density reaches a maximum around age 40 to 50, decreases with accelerating rate after age 50. So what the Neuro MRI NODDI analysis does, all these brain regions, we can calculate the total 5 NODDI variables in it. So 98 cortical gray matter volumes, 19 subcortical gray matter volumes, 34 cerebellum volumes, 53 white matter volumes, and combined volumes. In total, we get 5 NODDI variables are measured in over 240 volumetric regions of interest. The only areas that we don't actually measure NODDI in is impure cerebrospinal fluid, such as your ventricles, for example. So that is the density in the physical structure voxel by voxel, which is what the NODDI analysis allows us to do. The fourth part of understanding the brain as it ages and as it structurally is defined is the disconnecting brain. So how we measure the disconnecting brain is that we look at white matter fiber densities, and we look at the fractional anisotropy maps of how each brain region is connected to another brain region. And again, this is another variable that reproducibly decreases with age. So as we get older, the entire white matter connectivity. This is not a gross measure of white matter volume. This is not like volumetrics where you're just trying to understand gray matter versus white matter. This is actually measuring tractography of the human brain. And it's showing that the actual tractography of the human brain reproducibly decreases with age. And what we measure with the Neuro MRI is called a connectome. And we start with an atlas of 145 brain regions. And for each of these 145 regions, we look at how strongly they are connected to the other 144 regions. So each row of this connectivity diagram measures the connectivity across all 144 regions. And so if you take a look at this as the left hippocampal region, this will tell us which physical brain structures this left hippocampus is connected to and the strength of that connection. And this is a quantitative measurement. So we can measure its change with time and with intervention. So in summary, we get 145 by 145 gray matter connectivities measured. It's quantitative individual region to region analysis, total connectivity for each region measured. So these are the 4 real core areas of physical structure that you can't make up. It's not a functional, it's actually what is the physical structure of that human brain with disease or with age. volumes, cortical thickness, neurite density, the NODDI analysis, and the white matter connectome are the 4 core components of brain structure and health. So what do we do about it? Are we just watching your brain shrink and watching you die, or can we actually physically do something about this? And that's why the Neuro Restore was created, because we can start restoring this shrinking, denervating brain using advanced nutrition. What Neuro Restore does is it combines all the core components of brain biochemical and physiological health required for brain maintenance and brain growth, if you will. Restoring and creating plasmalogen reserves, restoring and creating these methyltransferase reserves, mitochondrial system, peroxisomal system, cholesterol metabolism and transport. We often forget about brain and muscle energy support, and the protocol deals with that, immune support, and the micronutrient support. And if you look at these core systems and you apply them, you can make dramatic impacts on the human brain. So what I want to do as an example, and using myself as an example, okay? Thinking that I was going to be a control person, turns out that I wasn't such a good control. So I had a cement fall I fell on back of my head in 1977, about age of 8. And, I was in the hospital for about a week, but I was a young kid. They sent me home and said, live your life. And I've lived a pretty decent life. And so at age 53, I was healthy, symptom-free, high-functioning guy running a company, figured, hey, what's wrong with me? So I get my first MRI in 2022 at age 53 and started the Neuro Restore protocol. And I've got a second one at age 54, third one at age 55, and I'll be getting a fourth one here in the next month or so. And let's see what happens when we actually target human brain physiology. First thing is restoring brain volume. And this case study is important because this is what you're going to deal with your clients and your patients, because they're going to get access to certain MRI technologies, most commonly volumetrics. And they're going to look at, oh, the volume of my hippocampus is associated with this, or my volume of my frontal cortex. And they're going to get all worried about what a volumetric analysis is going to have. And you're going to be stuck trying to interpret this information for your clients. So let's talk about the brain volume, which is kind of the basic, most simplest part of MRI analysis of the brain. It's still important, okay, because your brain does shrink, but you have to make sure we interpret it properly. So this is my brain at age 53 and at age 55 and a half. Okay, so using the Neuro Restore platform. So first and foremost, my whole brain volume has not changed. If not, my entire brain volume has gone up a little bit. But there's been a very significant increase in my gray matter volumes. My ventricular volumes have stayed constant, which is good. Whole CSF has gone up slightly, but basically constant. But there's been this change in my white matter volumes. This is a classic, called a T1 MRI scan. And so I have this reduction in white matter volume, well, which on first, thought, you say, well, what's going on with my white matter? And so stable brain, increased gray, decreased white matter. So what's up? Is this a good or bad thing? Is my brain getting worse or better? You can't really tell from a volumetric analysis. Now, if I start looking at my cortical regions, okay, and you'll see here, generally I have smaller than average in certain areas. Like my frontal cortex volumes on average were relatively small versus the general population. This is why you want to really beware, because as a executive functioning person, I'm not usually quite limited in my executive functioning skills. And so where your frontal cortex is typically a measure of your executive functioning capabilities. This is where you want to be aware. Interpersonal brain volume variability is very, very high. Your brain is personally designed for you. It's been built based upon all of your living experiences. And so although on large populations, brain volumes are very good measures of certain things, at an individual level, you always have to be super careful. So interpret these personal cross-sectional comparisons to population averages very, very cautiously. But what you will see is that regardless of what I started at, generally speaking, most of my brain volumes have increased quite significantly. And the opposite is supposed to happen, right? Remember, I'm supposed to be losing 0.3 to 0.5% per year. So I should have lost over a percent of brain volume in this 2.5 years. If we look at the subcortical regions, which is where these cell bodies are, like your nucleus basalis, and so on and so forth. The basal forebrain, which is your cholinergic system, which is critical for, well, aging, your hypothalamus, but mostly people consider it associated with cognition. So acetylcholine is for cognitive functions. Those were dramatically increased. And you'll see some going up and some going down. What is interesting is my hippocampus, which most people get obsessed over, has gone down. Both my right and my left hippocampus volumes have decreased. So if this is the only information I had to go on, I'd be kind of freaked out by this. This is like, oh my goodness, my hippocampus is shrinking. That's not a good thing. That's where memory comes in. That's where all these things are going on. And so I want to know, what does this mean? And my white matter volumes across the board, basically, all of my white matter volumes are decreasing, other than my corpus callosum, which was small to begin with. And so my smaller corpus callosum is also a symptom of long-term head injury. Autistic children and head injury individuals over long periods of time will often have, especially if it happens early in your life. And my head injury occurred when I was young, when my brain wasn't fully developed. The point, though, is my brain aging at some sort of accelerated rate based upon this information? And this is where the caution comes in. And this is really the problem with a lot of the commercially available MRI that's being sold to people on a mass scale. The risk of misinterpreting this data is very, very high. Brain volumes are very crude measurements of brain health and should never be used in isolation. Neuro MRI is the only technology that can accurately interpret brain volume data. So stay tuned for what that means. So next thing, we talk about cortical thickness. So I showed you before how the cortical thickness was associated with cognitive functioning in those publications. So contrary to the brain volume analysis, you can see quite dramatic improvements in my back of my head, which is my occipital cortex, from 2022 to 2023 to 2024. And this region at the top, which is my motor cortex region, also dramatically improving in their thickness regions. And what's important about Neuro MRI is that it provides quantitative information. So if I look at my total brain thickness from baseline at 2.38, which still is not bad, and quite frankly, an inflamed brain typically has slightly elevated brain thickness for a short period of time before you kind of run out of runway on it. So I was about average, a little over average on cortical thickness at baseline. But you saw these areas here where I've circled there. This is a histogram of, these are all regions that had very low, very thin brain in those regions. And after 2.5 years of the Neuro Restore platform, you see systematic improvement in brain thickness, which is the exact opposite of the traditional aging trajectory. So my brain is aging in reverse. It is getting younger with each subsequent year in terms of the cortical thickness. But what you can see is all these areas here in the histogram, 10 to 15 regions with thin cortices have come way down, and the overall shifting of the pattern. And we can look into these things in greater detail. Visually, you can see, the thickening of my motor cortex and this thickening of the occipital cortex from a visual perspective using this high-resolution surface map technology. But we want to look at this, overall. The brain is complex. It can be quite overwhelming. You think, well, 600 regions, this and that. So how do I organize this into such a way that you can kind of pinpoint something that's important for you and your client immediately? And so the cortical areas, thickness areas, those 600 brain regions are organized into 17 networks. And we can compare them to a population to get a Z-score to compare how, and the Z-score is not so important in its absolute value. It's more important in its relative information. And so what you can see here, at baseline, I had 5 regions of 5 networks that were below 0. So they were smaller than normal, both in my left hemisphere and my right hemisphere. But after 2.5 years on the Neuro Restore program, dramatic improvement. So these areas of cortical thinning have been completely restored into normality over that 2.5-year period. Now this allows us to look at this region 1 and region 12. And so each of these networks are broken apart. So what's interesting is region 1 is my central visual area, and my Z-scores went from -0.9 or -1.6 to plus 1.2 or plus 0.2. And the other area, which is 12, is my peripheral vision. So my peripheral vision area also dramatically improved. So you can dig deeper and deeper into these situations. And as you go into sections 1 and sections 12, they get further broken down into what part of the brain are we talking about. And this is the extrastriate cortex, for example. And you can get deeper and deeper into the full network. So the central visual, you can see the z-scores here. So everything on the left of the line is low, everything on the right of the line is high. So you can see across the board, all of these regions are way below normal. And then 2.5 years later, they're all on the positive side. So we're at the point now where we can actually directly interact with the human brain and actually do something about what we see. And this is the peripheral vision. You see very similar aspects of it. So you can dig deeper and deeper and deeper. But the next section is really the big important part. This NODDI analysis and neurite density really is the critical component of understanding how the brain is physically organizing itself with age. So we showed you before this intraneurite water, how it very precipitously starts declining after age 50. Well, this is the intraneurite restoration with Neuro Restore. From across the board, from cerebral white matter, all my white matter tracts, okay, the fiber density of all my white matter tracts have dramatically increased over that 2.5-year period. And this free water, okay, which is the other component of the NODDI analysis, has gone down for the most part, and it's supposed to go up. So from a NODDI perspective, the variables of NODDI are moving in the reverse aging direction. So in terms of intraneurite in water, my brain is getting younger with each successive year. But the question is, there's areas that don't follow the same pattern. My cerebral white matter tracts, my right and left, both showed an increase in free water. And that typically is not what you want to see in the human brain. So we want to look into that a little more closely and say, can we understand that? Well, the third variable in the NODDI analysis is called extraneurite water. This is the water that is measured inside your microglia or your inflammatory cells. It's also in your healthy cells, like your neurons and your oligodendrocytes. But your inflammatory microglial cells can overwhelm this extraneurite water space. And what you're seeing here across all these white matter regions, dramatic reductions in this extraneurite water, which is associated with microglial inflammation. So when we look at these areas, if we take a look at this one white matter tract, which is the cerebral white matter tracts, okay, you can see the intraneurite water, which is a measure of axonal or myelinated axons and dendrites, dramatically increased in a 2.5-year period. The inflammatory component, which is all the microglial regions, have dramatically decreased. And there's been a slight increase in free water. So where is this free water coming from? Well, it's not coming from a loss of dendritic or axonal density, it's coming from a reduced level of inflammatory cells. And so increase in free water is not due to decreased fiber density, it's due to reduced inflammatory microglial cells. So interpreting this information is very simple and easy with Neuro MRI, because every single region, you get all those variables all in one spot. And so going back to this whole brain analysis, okay, we're looking at just the crude volumetrics. We can understand this crude volumetric data much more detail with confidence. We know what's going on now. So gray matter is going up because I'm increasing neurite density with the Neuro Restore platform. Total white matter volume is going down because I've reduced inflammation. Okay, so larger brain volumes aren't always good. Okay, autism, a diagnosing of autism is by measuring brain inflammation and brain swelling in children. So you can have brain swelling. Just because your brain is swollen doesn't mean it's a good thing. And so what we have here is this reduced white matter is really caused by this reduced level of inflammation. Or conversely, my high level of white matter was because I had an inflamed brain, which is consistent with a concussion or long-term concussion situation. So let's go talk about the hippocampus now. Again, this hippocampal volume has decreased, which would normally freak someone out. So we have large increases in the basal ganglia area, like the basal forebrain space, decreased hippocampal volume. If we look at the hippocampus in this NODDI model, which allows us to look at free water and tissue water, and then separate the tissue water into this intra and extra neurite water, it gives us a variable called the neurite density index, which is basically what proportion of the cellular water is in axons and dendrites relative to the total cell population. And then you'll see a lot of, if you're researching out, you'll see a lot of people using this NDI as a variable in publications. I personally like to look at all three separately. But now we take a look at the hippocampus and we say, okay, the total volume of my hippocampal regions in the 2.5 years has gone down. But the intraneurite density, so my myelinated axons in my hippocampus has gone up dramatically. Extraneurite water has gone down. So the overall neurite density, which is a measurement of the actual physical quality of my hippocampal space, has gone up dramatically, which is consistent with the other observations. So the final part is restoring the connectivity. Okay, so we talked about the volume of the brain, the thickness of this cortical band, and then we talked about the actual quality density of white matter and gray matter, what's actually in those three-dimensional cubes all across your brain. Well, your brain isn't a bowl of soup or isn't disconnected. So gray matter is connected to other gray matter. The hippocampus is connected to your amygdala. It's connected to your parahippocampus. It's connected to your other areas of your brain. So the question is how do we measure the physical connectivity of the human brain? And how we do that is with advanced tractography. Okay, and using advanced tractography with the NODDI, with diffusion tensor imaging, between 300,000 and 400,000 fibers, okay, are measured, okay, in the brain. And then these fibers, okay, are then organized into short and long paths. Like you can have short paths that happen just within, millimeters of each other, and you have long paths that'll go through your corpus callosum. And tractography organizes all of those little tracks into connectivities between one brain region to another brain region. And when you do that, you can take these 300 to 600,000 tractographies and you can organize them by actual brain regions of interest. Okay, and if we take these composite brain regions, if we look at the whole brain, we can ask the question, across the entire brain, what is the amount of myelinated fibers in that human brain? And so my total brain fiber levels have gone up, which is white matter, which is the myelinated axons. And it's across the board. My subcortical nuclei, right, left, the cerebral, cerebellar exterior, and so on and so forth. So all these major areas of interest, we see this increase in overall fiber density. In the cortex, as a general rule, major areas in the temporal cortex, my occipital cortex, which we saw increases in my cortical thickness areas, have dramatically improved. And in the subcortical regions, specifically this hippocampus, we talked about hippocampus before, but you can see the actual myelination, the innervation going into the hippocampus and the innervation leaving the hippocampus has dramatically improved. So my hippocampus is now more strongly connected to different regions of my brain than it was before. And the basal forebrain, which contains like the nucleus basalis and other cholinergic neuron cell bodies, that cholinergic system is now sending more axons to more cells in my brain. So finally, how do we report all this? How do we put all this in a way that you can actually quickly get to an answer and quickly get to some sort of action plan for your patient or your client? And so what we do is all of our data and the video is uploaded into Immorta. So we have this customized clinical trial software platform, but it is shareable and distributable. So this information, videos, and the data can be shared with your neurologist or anyone else you want to look at this information. We do create core high-level brain health summaries, And then we have detailed reports by individual regions of interest and consolidated regions of interest. And if you're interested in it, we can also create custom personalized regions of interest for longitudinal tracking. So for multiple sclerosis or some sort of brain lesion, we can identify that region and actually track that region over time. So the video, the T1, which is your high resolution volumetrics, both the axial, coronal, and sagittal views are provided in a video format that you can watch slice by slice. The T2 FSC is also done in axial, which is also pretty valuable. T2 flare, which is common for multiple sclerosis. T2 SWI, which is your susceptibility weighted, looks for brain bleeds, measures hemoglobin levels, both from the arterial and venous system of the brain. So these are all available for individuals. But this is what the ROI report looks like. So there's 253 ROIs and comprised of 142 pages. So I showed you the summaries of these areas, but now if I want to look at a particular region, you can see all of these variables for that region. So for the cerebral cortex as a whole, left versus right hemisphere, I can get all the T1 values, T2 hyperintensities, the arterial spin labeling, which is the blood flow, bold levels, DTI levels, NODDI levels, and fixal. I haven't gone through all these details, but they're there. So, but it allows you to take a look at one region at a time and look at the completion of that region. Cerebral blood flow is a critical measure of overall brain health and injury. It's a simple variable. The MS lesions are measured in T2 flare. SWT2 SWAN measures the arteries and veins. So all the things that a neurologist wants is still included, even though the Neuro MRI is designed for health optimization and reversing the aging of the human brain, which is more advanced than trying to understand if someone has a stroke or some sort of brain bleed. That's a classic neurologist to measure that in a relatively short period of time looking at this data. And so this is what a microbleed would look like in someone on the SWI imagery. And the videos are provided for all of this. The cortical thickness report is 21 pages, looks at the 600 brain regions, compares to a population distribution, and you can dig, simply deeper and deeper. The DTI connectome report is 145 pages, looks at each brain region and how it's connected. And it's actually indexed, so you can go from one region to another if you wish. BOLD measures the functional connectivity. Let's talk about functional MRI. And likewise, you get a 145-page BOLD report that looks at all the positive and negative active correlates per region of the brain. And then magnetic resonance spectroscopy, which I didn't get into today, also looks at 45 variables. And we measure two brain regions, the frontal cortex and the precuneus, and we can measure the actual biochemical composition of the brain. Glutathione levels, GABA levels, and so on and so forth. So overall sequence design, for those of you who want to understand the actual details, those who are happy, you can stop listening now if you want to. But the technical details are, we have structural brain analysis. We do a T1 high resolution, T2, FSC, T2 flare, T2 SWI, diffusion tensor imaging, NODDI, and fixal analysis. These are all part of brain analyses. In terms of structural brain analyses, in terms of functional brain analysis, we look at BOLD, arterial spin labeling, and magnetic resonance spectroscopy. All this is done within about 45 minutes to an hour in the scanner. Physical connectivity is this gray matter to gray matter connectome that I mentioned to you, DTI Tractography. It's 145 by 145 regions. And the functional connectivity is also gray matter to gray matter resting synchronous activity. So this is resting BOLD values, and then quantitatively measures the positive and negative functional connectivity. We also looked at vascular perfusion, which is the cerebral blood flow. Cerebrovascular reactivity, that'll be involved in future reporting, looks at respiratory challenge BOLD, and then the brain metabolic analysis with magnetic resonance spectroscopy. So in summary, the Biometrix Technology Suite is designed to be worked to work together. It's an integrated suite. It's been built over several years to do an integrated process. The Neuro MRI is the complete brain at your fingertips. It's the only way to optimize and track your brain health. If you're not measuring it, you really have no idea what's going on in your brain. The Bioscan blood test does the exact same counterpart, but for the biochemistry of your brain, all the biochemical systems of your body. It's a complete biochemical prodrome analysis of health, and it's the only way to track and optimize your biochemical health. There are markers in the Bioscan blood test that are not available anywhere else in the world. The complete plasmalogen analysis, the phosphocholine analysis, the sphingomyelins and ceramides, these all have meaning and are not available anywhere else but except in this Bioscan report. The Neuro Restore, again, we're talking about complete biochemical nutrition. And it is the only way to restore and optimize biochemical and physiological health. There are key components in this, like our animal-based phosphocholine system or the plasmalogen precursors that are only available through the Neuro Restore package. And they're designed for brain and physiological health, not just your brain, but your heart and your lungs, kidneys, and so on and so forth. At the end of the day, achieving perpetual health is not possible without this Biometrics Technology Suite. Without the Biometrics technology, you really don't know what's going on in your brain. You don't know what's going on with the biochemistry, and you have really no real way of intervening in creating the biochemical reserve capacity to stop the entropy of aging. Okay, it takes energy for us to reverse the aging process. And so reversing the aging process requires a very strategic metabolic engineering approach. And these three things work together for that. So thank you for your attention to this lecture on the Neuro MRI. Thank you.